Next, we will talk about the ABO blood groups. The ABO blood group system is a classic example of codominance. The alleles involved are IA, which produces antigen A, IB, which produces antigen B, and IO, often written as small I, which produces no antigen. IA and IB are codominant, meaning both are expressed together when present in the same individual. Both IA and IB are dominant over IO. In an individual with the genotype IA, IB, both A and B antigens are produced on the red blood cells, resulting in blood group AB. This AB is called codominance because both alleles are expressed equally in the phenotype. If both alleles of a gene pair are expressed equally in an individual, with neither being dominant over the other, they are called codominant. A well-known example of codominance is the human ABO blood group system, which includes 4 phenotypes, A, B, AB, and O. In this system, the alleles IA and IB are codominant. When an individual inherits IA and IB, their red blood cells express both A and B antigens, resulting in blood group AB. In contrast, both IA and IB are dominant over the allele IO. Individuals with blood group O, with genotype small ii, do not have A or B antigens on their red blood cells. Now let's look at this diagram, which illustrates the ABO blood group system. It classifies blood based on the presence or absence of specific antigens on the surface of red blood cells and antibodies in the plasma. Each blood group has a unique combination. In Group A, red blood cells have A antigens and the plasma contains anti-B antibodies. In Group B, red blood cells have B antigens and the plasma contains anti-A antibodies. In Group AB, both A and B antigens are present on the red blood cells, and there are no antibodies in the plasma. In Group O, there are no A or B antigens on the red blood cells, but both anti-A and anti-B antibodies are present in the plasma. Antigens are markers found on the surface of red blood cells. The body recognizes its own antigens as self, so they are not attacked by the immune system. Antibodies, on the other hand, are proteins found in the plasma that attack foreign antigens. For example, a person with Group A blood has anti-B antibodies, so if they receive blood with B antigens, their body will react against it. This leads to the concept of agglutination. If incompatible blood types are mixed, antibodies bind to foreign antigens, causing red blood cells to clump together. This can block blood vessels and is dangerous. Finally, in terms of blood transfusion, Group O is known as the universal donor because it has no A or B antigens, so it is less likely to be attacked by the recipient's antibodies. Group AB is known as the universal recipient because it has no anti-A or anti-B antibodies, so it can receive blood from any ABO group. The possible genotypes and phenotypes in the ABO blood group system can be explained clearly. Individuals with blood group A can have the genotype IA, or IA, small I, while individuals with blood group B can have IB, or IB, small I. Blood group AB has the genotype IAIB, and blood group O has the genotype both small II. Since IA and IB are dominant over small I, a person with blood group A may have either IA or IA small I, and a person with blood group B may have either IB or IB small I. In contrast, blood groups AB and O each have only one possible genotype. AB is always IAIB, and O is always small II. An interesting point is that blood group O can still appear in offspring even if neither parent shows it. For example, if one parent has blood group O with genotype IA small I and the other has blood group B with genotype IB small I, their children can inherit any of the 4 blood groups out of A, B, AB, or O, each with an equal probability of 1 in 4. So, in the ABO blood group system, a person's blood type is determined by the combination of alleles they inherit from their parents. Each individual receives one allele from each parent, and these alleles together form the genotype, which then determines the blood group, or phenotype. For blood group A person can have either 2 AA alleles or 1 A allele and 1 O allele. In both cases, the A antigen is produced, so the blood group appears as A. Similarly, for blood group B, a person can have either 2 BB alleles or 1 B allele and 1 O allele, and in both cases the B antigen is produced, giving blood group B. Blood group AB occurs when a person inherits 1 A allele and 1 B allele. In this case, both alleles are expressed equally, so both A and B antigens appear on the red blood cells. This is an example of codominance, where neither allele masks the other. Blood group O occurs when a person inherits 2 O alleles. Since the O allele does not produce any antigen, the red blood cells have neither A nor B antigens. Overall, this shows that different combinations of alleles result in different blood groups, and the dominant, recessive, and codominant relationships between alleles determine the final phenotype. In the cross, one parent has blood group A, with the genotype IA small io, and the other parent has blood group B with the genotype IBIO. Even though neither parent shows blood group O, both carry the recessive small I allele. During gamete formation, the parent with blood group A can pass on either IA or small I, while the parent with blood group B can pass on either IB or small I. When these gametes combine, 4 possible offspring genotypes can be formed. These are IA, IB, which results in blood group AB. Then, IA small I, which results in blood group A. Also, IB small I, which results in blood group B. And both small II, or IOIO, which is a recessive allele and results in blood group O. Each of these combinations has an equal chance of occurring, so the ratio is 1A to 1B to 1AB to 1O. This means there is a 25% chance of producing a child with blood group O, even though neither parent has blood group O as their phenotype. As shown here in the example. Here, one parent has blood group A, and the other has blood group B. To get all possible outcomes, we consider both parents as heterozygous. So, The parent with blood group A has the genotype Ia, small I, and the parent with blood group B has the genotype Ib, small I. During gamete formation, the parent with blood group A produces two types of gametes, Ia and small I. Similarly, the parent with blood group B produces two types of gametes, Ib and small I. When these gametes combine in a Punnett square, four possible genotypes are formed. These are Ia, Ib, Ia small I, Ib small I, and both small ii. Each of these genotypes results in a different phenotype. Ia, Ib gives blood group AB, Ia, small I gives blood group A, Ib small I gives blood group B, and both recessive small ii gives blood group O. All combinations are equally likely, so each occurs with a probability of 25%. This gives a genotypic ratio of 1 Ia Ib to 1 Ia small I to 1 Ib small I to 1 small ii. In terms of phenotype, the ratio is 1A to 1B to 1AB to 1O. This example clearly shows that even if neither parent has blood group O, it can still appear in the offspring because both parents carry the recessive small I allele.